All right, today we're tackling something huge. We are going to decode the language of life itself. In this explainer, we're diving straight into bioinformatics, this amazing field that mashes up biology, computer science, and just massive amounts of data to unlock the secrets hidden deep inside our DNA. So, if you've ever glanced at a course outline for bioinformatics, you know it can look, well, a little terrifying. All that jargon, the complex terms, it's enough to make your head spin. But trust me, stick with me for just a few minutes, and I promise we're going to slice right through that noise and make total sense of the big ideas. You know, the reason this whole field even exists boils down to one simple fact. Modern biology has a data problem. And I mean a massive data problem. Every single day, labs all over the world are generating this absolute tidal wave of information from sequencing genomes. The real question is, what on earth do we do with all of it? And that question leads us right into our first section. The answer to the data problem? It's bioinformatics. It's the essential bridge between the science happening in the lab and the power of the digital world. Okay, so what is it really? At its heart, the definition is pretty simple. It's the science of using computers to collect, store, and most importantly, analyze all that complicated biological data. Just think of it as a super powerful toolkit for biologists, letting them handle information that is just way too big for any single person to wrap their head around. So when we say data, what are we actually talking about? Well, it's basically the code of life, but turned into digital files. We're looking at DNA sequences, that's the master blueprint for everything in a cell. We look at RNA sequences, which are kind of like the messengers carrying instructions from that blueprint. And we study proteins, the actual workers that do all the jobs, both as simple strings of text and as the complex 3D machines they fold into. Okay, so we've got all this data digitized. Great. But before you can do any of the cool analysis, you have to get organized. And this is where the whole idea of biological databases comes into play. Seriously, imagine trying to find one specific book in a library where millions of books are just dumped in a giant pile on the floor. It would be completely impossible, right? Well, that's what databases are for. They are these massive digital libraries that catalog all the building blocks of life, giving them structure and making them usable for scientists anywhere in the world. And these digital libraries, they basically come in two main flavors. First, you have your primary databases, like the famous GenBank. Think of these as just raw data archives. A scientist sequences something, they upload it, it's just the raw text. But then you have secondary databases, like Uniprot. These are more like curated encyclopedias. They don't just have the sequence, they've got notes from experts explaining what it all means, like, hey, this part does this job, or a typo right here is linked to a certain disease. Right, so everything's neatly filed away in these digital libraries. But now there's a new problem. How do you find the one specific gene you're looking for when there are millions or even billions of other entries? The answer? You use a specialized search engine. And the most famous tool for this, the one you'll see on every single bioinformatics syllabus, is called BLAST. The absolute easiest way to think about it is, it's Google, but for genes. You paste in a sequence, hit search, and it scours the world's biological databases to find things that look like it. And how it works is just beautifully simple. First, you give it a sequence you're interested in, then BLAST takes that sequence and just blitzes through a gigantic database like GenBank at incredible speeds. And here's the crucial part. It's not just looking for a perfect match. It's smart enough to find similar sequences, which is key because of all the tiny changes that happen during evolution. Finally, it spits out a ranked list of results, your hits, showing you which known genes from other creatures are the closest match to yours. But finding a match is really just step one. The real magic, the part where the actual scientific discoveries happen, is when you take these sequences, put them side by side, and start comparing them. This whole process is called sequence alignment, and honestly, it's probably the most fundamental technique in all of bioinformatics. By lining up two or more sequences, whether it's DNA or protein, we can see exactly where they're identical and where they're different. And it's those similarities and differences that tell us these incredibly powerful stories about how things work and where they came from. Now, the important thing to know is that there are different ways to compare. If you've got two genes that you suspect are very closely related, like they're basically cousins, you'd use a global alignment that tries to match them up from beginning to end. But what if you're looking for just one small important pattern that might be shared between two totally different proteins? Well, then you'd use a local alignment to hunt down just that one little region of best fit similarity. Okay, so we can store the data, we can search the data, and we can compare the data. But that brings us to the biggest question of all. So what? what's the point? What are the truly profound, big picture stories we can uncover by doing all this? And this is where it all comes together. This is the payoff for all those other techniques. We get to map the history of life itself through something called phylogenetic analysis. The core idea here is it's beautiful. It's so simple. The more similar the DNA is between two different creatures, the more closely related they probably are. And if we apply that simple logic across thousands of different species, we can use all those sequence alignments to build a gigantic family tree that shows how every living thing on this planet is connected. And what this reveals is just mind-blowing. These trees can show us the evolutionary journey from ancient bacteria all the way to us. They can help us track the origin of a virus, like COVID-19, and literally map its spread across the globe. They help us watch how critical proteins have changed over millions of years, and even let us trace our own genetic story all the way back to our most ancient common ancestors. So, why does any of this matter in the real world? Well, in medicine, this is how we find the genetic cause of a disease and start designing drugs to fix it. In basic research, it's how we unlock the deepest secrets of how life even works. And in diagnostics, it's the engine behind all those fast and accurate tests that can spot an infection or a genetic condition. It's a field that is completely changing how we think about health and life itself. We've built the libraries, we've designed the search engines, and we have started to read the book of life. But the truth is, we are still on the first few pages. And the most exciting part, the thing that drives this entire field, is that one big question. What other incredible secrets are still locked away in our DNA, just waiting for the next tool, the next idea, to finally decode them?